So previously I went through what were called the light-dependent reactions. So these are the reactions where with photosystem 1 and photosystem 2, we made ATP and NADPH. Technically, photosystem 1 passes on electrons to NADPH, and photosystem 2 passes on electrons to an electron transport chain that increases the concentration of hydrogen ions, which can ultimately be used to make ATP. So we get the energy from light to excite electrons, and that energy is then used to make these molecules. So now that energy is stored there. So the energy from the sun is stored in NADPH and ATP. That's happening in the chloroplast, and that's all happening at the innermost thylakoid membranes. What we're going to talk about now are the light-independent reactions. Okay, the light-independent reactions are going to happen here in the stroma, inside the chloroplast, but in the area outside of the thylakoids. Now, what's going to happen in the light-independent reactions? Well, we're going to get carbon dioxide. I lost my color here. There we go. Carbon dioxide. And it's not turning out so well. And a 5-carbon molecule called RUBP, which we'll get to in a second. It stands for ribulose sugar bisphosphate. So it's a 5-carbon molecule, 5-carbon sugar with a phosphate on each end. We'll look at that. We're going to use ATP and NADPH, and we're going to end up producing as products G3P, which is glyceraldehyde 3-phosphate. Again, we'll write that down, and we'll go over what it is. And we're going to make more of the RUBP. That's essentially what happens. Now, it's not a balanced equation. We'll see. We'll get the numbers. We'll put the numbers in here, and you can actually see how it actually happens. But that's the overview, the idea of what's going to happen in the light-independent reactions. We're going to get the energy from the ATP and NADPH to pull the carbon dioxide from the atmosphere. So this is called carbon fixation. It's going to bind that to the RUBP. Then they're going to split, and then we're going to run through a process that we're going to talk about called the Calvin cycle or Calvin-Benson cycle, which is also referred to as light-independent reactions. And I'll also emphasize here why they're called independent is because people used to refer to them as the dark reactions. So we have the light reactions and the dark reactions. The light reactions are dependent upon light. Without light, then they stop. We don't make any more ATP or NADPH. However, the light-independent reactions, while they can happen in the dark, they also happen in the light. They are dependent upon ATP and NADPH. So once you have those, then it can run. So it can run while it's light, and it can run while it's dark. So we'll keep those things in mind. So here in the stroma, what we're going to have happen is carbon dioxide is going to come in, being taken up by the plant, and it's going to bind to an enzyme called, well, its abbreviation for it is RubisCo. So it sounds like a funny name. It's ribulose bisphosphate, and then carboxylase. So RubisCo, it's kind of where that shortened name comes from. So this is an enzyme. We find it in any organism that is photosynthetic. So within the cells of the organisms that are photosynthetic, we find it. This is the enzyme that carries out carbon fixation. This is the enzyme that removes carbon dioxide from the atmosphere. So when we say plants take up carbon dioxide, this is the enzyme that's actually doing that. Okay, so this is the protein that's actually binding the carbon dioxide and pulling it out of the atmosphere. It's going to then bind to carbon dioxide and combine it with a molecule that has 5, 1, 2, 3, 4, 5 carbons, and then a phosphate on each end. Okay, that's the RUBP, or ribulose bisphosphate. So that's that molecule here. And then the enzyme, the carboxylase, adds the carbon or the carbon dioxide to this. You would think the result would be a 6-carbon molecule then. So this is actually very similar to the beginning of the citric acid cycle, if you recall. All right, we get a molecule there, but then it's a 4-carbon molecule, the oxaloacetate. It binds to the 2-carbon, the acetyl group of the acetyl-CoA, and then it makes a 6-carbon citric acid for the citric acid cycle. This is different, but you can see there's some similarities. How it's especially different is that when these two bind, only very temporarily are they actually together as a whole molecule. It almost immediately splits into 2 3-carbon molecules, each with all phosphates. And those are called 3 phosphoglycerates. PHOSPHOGLYC. 3 phosphoglycerate. So this is the 3, 1, 2, 3 carbon, and you have the phosphate there. Now, before we go any further, I'm going to have to clarify a little bit more about what's actually happening. When this process occurs, it does not happen with 1 carbon dioxide at a time and 1 RUBP. There's actually going to be 3 carbon dioxides coming in here, and they'll be joining with 3 RUBPs. Okay. So that means there's going to be 3 sets of 2. Okay, so we're actually going to have 9, so 1, 2, 3, 4, 5, 6. We're going to have 6 of these. So we're going to have this one gets split, a second one will be split, a third one will be split, a fourth one will be split, the fifth one will be split, and then the 6th one will be split. All right, so this is what we're going to have here. Now, so this is kind of like from joining 1 of these together, 2 of these together, 3 of these together. Okay. So they're all going to be 3 phosphoglycerates, so they're all going to have, a phosphate on the end, like this. Now, what's going to happen is these molecules are all going to become rearranged into a slightly different 3-carbon molecule. And with the phosphate on it here. That is G3P. So it's going to be glyceraldehyde. Phosphate. Now this is going to take up, so this part of the process here is going to require 6 NADPH and 6 ATPs for this to occur. All right. So what's happened? We bring in carbon dioxide. It joins to a molecule called RUBP. They split. This happens with 3 separate carbon dioxides and 3 separate RUBPs, right, to make up all 6 of these 3 phosphoglycerates, and they turn into 6 glyceraldehyde 3-phosphates. There's 6 of those. Now, 1 glyceraldehyde 3-phosphate, 1 of the 6, leaves. So we said here in this little equation, I said carbon dioxide and RUBP plus ATP and an ADPH is going to give us a G3P and then more RubisCo. So there's actually only going to be 1 G3P manufactured and put out. That's the one product that leaves the light-independent reactions. So G3P. That's the product, and only one of those. And just as a side note, I'm not drawing there's oxygens and hydrogens attached to these carbons, and you can see that I draw the structure of it when I draw glycolysis and parts of pyruvate oxidation and some other processes as well. And you could look it up, but I'm just trying to simplify it here. We're just not drawing the hydrogens and oxygens, just so you know there would be additional hydrogens and oxygens here. But this is generally the product, a 3-carbon molecule with a phosphate. That's it. You don't produce fructose or glucose or sugar or any other sugars. That's not what happens here. So when I said earlier on that carbon dioxide uses light, and there was that equation that people have with like water plus carbon dioxide plus light equaled these 6-carbon sugars, that doesn't happen. The product is a 3-carbon molecule that's a precursor to the sugars. That can then go through gluconeogenesis and become glucose, and then be stored as starch and other things. But in this case, this is the actual product. Now, what do you have left? You have 5 remaining G3Ps. There are going to be additional ATPs used. So 3 more ATP are going to be used. And those 3 ATP are going to turn these 5 G3Ps into 3 RUBPs. So you might be thinking, well, what? You know, like that you're a little bit lost on what I'm saying here. Okay, so I'll just try to sketch it out over here and simplify. We're not going to go through all the biochemical steps, but just sort of the overview of this. All right. So what you're going to have are 1, 2, 3, 4, 5 molecules. They each have the phosphates. Each of them have 3 carbons, right? 1, 2, 3. Okay. So there's 5 of these. There's 1, 2, 3, 4, 5. So that's 15 total carbons. Okay. Now what's going to happen is they are going to be rearranged through a series of chemical reactions into 3 of the RUBPs. So you're going to end up with 1, 2, 3, 4, 5, 1, 2, 5, 5. A phosphate now on each end. Okay, so you have 1 RUBP, 2 RUBPs, 3 RUBPs. All right, that's what's going to be made here. There are 15 carbons here, 5, 10, 15 carbons there. So these same 15 carbons get rearranged back into RUBP, right? And it takes extra ATP to do that. So kind of to add in here, this process is going to take the 6 ATP here to make the G3Ps, and then 3 more, so it's going to take 9 ATP in order to do this. But just these, and then these 6 NADPH. So you have 4. In this case, we have 3 carbon dioxides coming in with 3 RUBPs. That's going to require the use of 9 ATP, 6 NADPH, still just to make 1 G3P, and then back to getting the 3 more RUBPs, which are these same ones. Okay. So this is a cycle. All right, this is called the Calvin-Benson cycle. Because, like the citric acid cycle, the cycle produces at the very end one of its own initial substrates. So RUBP is made by the process, and RUBP is used right at the very beginning of the process. Those are what are referred to as the light-independent reactions, also called the Calvin-Benson cycle. So this equation here is one thing you should know. You should be able to cite, how many ATP are required during the Calvin-Benson cycle, how many NADPH are required for the Calvin-Benson cycle, so on, that type of thing. What's a product of the Calvin-Benson cycle? The really thing that leaves, that goes to the cell uses, is the one molecule of G3P, that one 3-carbon molecule with a phosphate on it. The other product, which is the RUBP, doesn't really go to the cell. It's recycled back into this process itself, right? And this is the process that removes carbon dioxide from the atmosphere. So this is where carbon fixation occurs. And remember, the name of the enzyme who removes the carbon dioxide from the atmosphere is ribulose bisphosphate carboxylase, which we also call RubisCo. All right. As far as the details of this, like the actual chemical reactions and how it's arranged in the intermediate molecules, it's not something that you need to know in our class. It's something that at one point, as I was looking this up to see, I want to see all the steps and all, it's very difficult to even find. I looked in biochemistry textbooks and botany textbooks and other sorts of books just to find it, and I really had to look in just peer-reviewed publications to see some of the steps of the process. It's not something that's commonly seen in many undergraduate texts of any kind at all. So it's not something for you to be particularly worried about. You just need to worry about the numbers, the names of the molecules, and the overall process. And you should be good with it. All right. So now, putting this all together with photosynthesis, we've used the energy of light, right, to make organic molecules. And really, the organic molecule that we've made is this G3P. That's really the actual product at the very end. And then that can be used in a separate process to make the sugars, all right, that then get stored in the cells, that then can be used by the plant or the photosynthetic organism, and then is the nutrients for other organisms that eat them. They eat plants and eat algae and eat those other things. So that's how the energy from the sun comes to all of us, essentially, in a way, through this process. Right? So just make sure you know names, numbers, and some of these details that I talked about, and you should be good.